So far, we've talked in detail about skeletal muscle. Let's now shift our attention back to the other two categories. We've seen this table before. Skeletal muscle is connected to your bones, your skeleton. Cardiac muscle is in the heart. Smooth muscle wraps around your internal organs. Cardiac and smooth muscle are controlled by the autonomic nervous system. You don't have conscious control. And cardiac muscle is similar, roughly, to skeletal muscle in the way that it looks under a microscope. It has sarcomeres, which create a cross-striated pattern, whereas smooth muscle has a much more diffuse appearance. Here's the question. How do cardiac and smooth muscles function differently? That is, what goes on within the interior of those cells that explains the differences in their functional properties. So let's start with cardiac muscle. Here we have a schematic. It looks pretty similar to a skeletal muscle, but what you might notice up above are these gap junctions. Gap junctions electrically couple cells together. All right, we see one right there. What that means is the cell that's sort of, we only see the little bit of it up above. Okay, this cell up here. Whenever it depolarizes, then that depolarization is going to follow through. So the gap junctions allow for ion flow. So when there's a depolarization of the upper cell, then it's going to serve to depolarize the lower cell. Let's step through the excitation contraction coupling of a cardiac muscle and see how it's different from a skeletal muscle. First of all, those gap junctions are a point of distinction. So an action potential travels between neighboring cells via those gap junctions. That includes neighboring contractile cells. So up above, we have a depolarization, which we see in red there. That will just automatically produce a depolarization in the cell that we're focused on, including through the T-tubules, which are similar to the T-tubules in skeletal muscle that we have in cardiac muscle. Another difference, though, are the receptors within the T-tubules. So instead of the DHP receptors that we saw in skeletal muscle, the T-tubules in cardiac muscle are packed with voltage-gated calcium channels. So when that action potential propagates through the T-tubule, it will activate those calcium voltage-gated channels that allows for a flooding into the interior of the cell of calcium. Now, intracellular calcium will just, diffuse in all directions. It can act directly on the sarcomeres, so it will bind to troponin. We've got tropomyosin in there. All of the sarcomeres function in a similar way for cardiac muscle as skeletal muscle. But that rise in intracellular calcium will also serve to activate the SR. The sarcoplasmic reticulum in a cardiac muscle cell also has ryanodine receptors, but they're of a different type. These ryanodine receptors are activated by intracellular calcium. So we have calcium activating the RYR, which in turn increases intracellular calcium. Then you've got the potential for a positive feedback loop where the calcium from the SR will then activate other ryanodine receptors to then release more calcium. We also have a pumping of calcium back into the SR as we saw for skeletal muscle. So all of the other events in the EC coupling are the same or similar enough anyway in the cardiac and skeletal muscle. So all what goes on in the sarcomere, the recycling of calcium back into the SR, that's all similar. But what we have here are the salient differences highlighted in this schematic. Now, another difference between skeletal and cardiac muscle is that the twitch tension can vary in cardiac muscle. Skeletal muscle, it's conserved. You've got essentially a fixed unit of force in skeletal muscle, whereas the amount of tension in a cardiac muscle can vary. So unlike skeletal muscle, twitch tension, so cardiac muscle cells are also called myocardial contractile cells. So a myocardial cell can be varied. And that's achieved by a variable concentration of calcium. So within a twitch, you can have different levels of calcium inside the cell. Now, the reason why and the mechanism that creates that variable calcium, we're going to get to later. All I want you to know is that this is one important difference between cardiac muscle and skeletal muscle. Let's talk about smooth muscle. We've seen in skeletal muscle, we have the DHP receptor activating through a mechanical link, the ryanodine receptor, which triggers a release of calcium. Cardiac muscle is a little different. The ryanodine receptor is activated by calcium. But in both cases, that increase in intracellular calcium does the same thing. It activates troponin, it moves tropomyosin out of the way. That allows the myosin cycle to occur. In smooth muscle, the thin filaments exist, but they don't have troponin. They don't have tropomyosin. So the whole mechanism for controlling tension generation in skeletal and cardiac muscle just does not exist in smooth muscle. Calcium still matters, but it acts differently on the thick and thin filaments. Intracellular calcium, as it rises in a smooth muscle cell, will activate an enzyme. That enzyme is known as calmodulin. Calmodulin, once it's activated, can then activate myosin. Okay, so myosin in a smooth muscle is of a different type than in the cardiac and skeletal muscle. It's a little bit more intelligent because it is interfacing, its generation of tension is directly controlled through calmodulin, but it's unlike skeletal and cardiac muscle where the myosin will just, as soon as there's any binding site, it will just ratchet along the thin filament. In this case, the activated calmodulin is necessary in order for that to occur. So you still have this rise in intracellular calcium causing an increase in force. It's just through a different mechanism. Now, something about smooth muscle that can be infuriating for an introductory instructor is just how variable they are. For example, some smooth muscle cells have an SR. So we'll say in some, you have an activation of calmodulin through the calcium that comes from the SR. But in other cases, the calcium comes from the interstitial fluid that is outside the cell. So if there's a cell membrane, there are calcium channels, and when they are activated, they allow for an increase in intracellular calcium. So intracellular calcium still matters. It can come from different sources depending on the type of smooth muscle. And of course, you can have a combination like the cardiac muscle. Calcium doesn't know where it's going. It's just randomly diffusing into the interior of the cell. So even if there is an SR, any particular calcium ion might activate the calmodulin directly. So we've got this rise in intracellular calcium through an action potential across the cell membrane, potentially through ligand-gated channels. We do not see that in this schematic, but that's another means. And then there are stretch-activated or mechanically gated channels as well. So if the cell membrane is pulled on, that can elevate intracellular calcium by allowing calcium across the membrane. So mechanically or stretch-activated channels are another means for elevating calcium. And whenever calcium goes up, you should expect more force to be generated through the activated calmodulin. All right, so we can see that things are pretty different in smooth muscle and variable among smooth muscles. Smooth muscles are also interesting in that they have properties that are unlike skeletal and cardiac muscle beyond just the calcium flux. For example, some smooth muscles can undergo what's called a latch state. Now, we already talked about rigor mortis and how the binding of myosin heads to those G-actin binding sites in the thin filaments can provide a very rigid connection to make the tissue quite stiff. Smooth muscles are capable of a similar level of stiffness when they go into this latch state. Now, the way that that's achieved is by depressing intracellular calcium. And that serves to, just like we saw in the myosin cycle for skeletal muscles, maintain the binding between the thick and the thin filaments. So smooth muscles take advantage of that to essentially go into a state where they become a rigid block of tissue that doesn't require any ATP. Later on, we're going to talk about the circulatory system. We have arterioles. They are blood vessels that are wrapped in smooth muscle. And the diameter of those arterioles really matter to regulating blood pressure and the shunting of blood into different regions of the body. Because those blood vessels are using smooth muscle, it requires very little energy for them to maintain their diameter. In contrast to, say, a sphincter that has skeletal muscles, which requires ATP to maintain tension. So the latch state is a pretty neat trick. Smooth muscles are also capable of changing membrane potentials autonomously without any inputs from, say, a parasympathetic neuron or any kind of release of neurotransmitter. These cells by themselves can generate the excitation that drives contraction just spontaneously. So they're really weird if you're used to thinking about skeletal muscles. Now, another important difference has to do with this business of cross-striated versus a diffuse appearance under a light microscope. That turns out to have functional properties. So in skeletal and cardiac muscles, we've got this crisscross arrangement. The thick and thin filaments are aligned, but under a microscope, it looks like a crisscross pattern. Smooth muscles don't have sarcomeres. But you do have thick and thin filaments oriented in a variety of directions. Watch this. What happens to a smooth muscle as those myosin heads ratchet along the thin filaments? We can see, because we don't have this structured arrangement of the thick and thin filaments, unlike what we see up above, where there is, within a sarcomere, no overlap between the thin filaments on this side and that side. We have no filament overlap there. When the smooth muscles contract, it allows for that overlap in thin filaments. As a consequence, the thick and thin filaments can contract to a greater extent because you essentially don't have that interference between the thick and thin filaments or neighboring sarcomeres, like we saw when we were talking about the length-tension relationship for skeletal muscle. We don't have that here for smooth muscle. Instead, the thick filaments are free to step along the thin filaments to a greater extent. And that allows for greater changes in length of the muscle than you see in the skeletal and cardiac muscle. As a consequence of not having all of your myofibrils and your muscle fibers oriented in the same way as you see in a lot of skeletal and cardiac muscle, where you get tension only in one direction, smooth muscle can be arranged in sheets that have fibers oriented in a variety of directions. So you can generate tension along a plane in more than one direction.